Have you ever stopped to think about the single greatest engineering challenge that every single living thing on this planet has solved? Seriously. It's this incredible puzzle of building something that can last a lifetime, but at the same time, it has to be flexible enough to react in a split second. It's like building a fortress that's also a dance floor, all at the molecular level. So how in the world does life pull this off? So here's the fundamental paradox of life. On one hand, you need incredible stability. I mean, think about your DNA. It has to preserve your genetic blueprint with absolute rock-solid integrity for decades. But on the other hand, life has to be intensely dynamic. Proteins, for example, have to bind, release, and totally change their shape in milliseconds just to respond to what's going on. So you've got this need for permanence and this need for constant change. How can those two opposing needs possibly exist in the same system? So, yeah, that's the big question we're tackling today. How does life build structures that are unbelievably stable, yet at the same time allow for all the constant dynamic interactions that actually, define being alive? And the answer? Well, it turns out to be just beautiful in its simplicity. See, nature doesn't use one tool for this job. It uses two, and they're perfectly balanced against each other. The entire solution lies in this hierarchy of two totally different classes of molecular forces. You've got the super strong covalent bonds to build the permanent framework, and then you have these weak non-covalent interactions to handle all the dynamic stuff. All right, first up, let's look at that covalent blueprint. These are the bonds that give life its permanent, unchangeable structure. So what is a covalent bond? Well, it's formed when two atoms literally share a pair of electrons. You can think of it like a molecular handshake that's been sealed with superglue. It is incredibly strong and, for all intents and purposes, permanent. To change a molecule's basic structure, you have to chemically break these bonds, and that takes a ton of energy. This is what links together the backbone of our proteins and our DNA, creating that permanent, unchanging scaffold of life. And wow, this chart really puts their strength into perspective. See that tiny little sliver on the left? That's the random thermal energy that's just bouncing around inside a cell. Now compare that to the massive amount of energy you'd need to break a covalent bond. It's not even a fair fight. They are literally orders of magnitude stronger, which is why the covalent framework of our molecules is completely stable and totally unfazed by the chaos of the cell's environment. But a permanent scaffold is just a statue, right? It's not alive. To bring it to life, we need the second part of the solution, these non-covalent interactions. And you can think of these as the dynamic language of life. Now, unlike their covalent cousins, these interactions are, individually, very weak. They're constantly breaking and reforming. But here's the real genius. It's all in the numbers. Stability here doesn't come from a few super strong links, but from the cumulative effect of thousands, even millions of these weak ones working together. This collective power has a name. It's called cooperativity. So you might be asking, what exactly are the words in this dynamic language? Well, there are four main types, and they basically orchestrate almost everything that happens inside a cell. First, you've got hydrogen bonds. These act like tiny molecular architects because they're very specific about the direction they point in. Then there are ionic interactions, which are kind of like a context-dependent glue. They can be strong or weak depending on what's around them. Third, we have van der Waals interactions. This is a universal but very short-range attraction between pretty much any two atoms that get close enough. And finally, my favorite, the hydrophobic effect. This one isn't really a bond at all, but it's an incredibly powerful organizing principle. It's basically driven by the fact that water molecules would much rather stick to each other than make room for oily molecules, so they effectively push all those oily parts together. And this slide just illustrates the power of cooperativity perfectly. A single one of these interactions, it's fragile. It's easily broken by the random thermal energy in the cell. But, and this is the key, when you have thousands of them all lined up between two big molecules, the whole structure becomes incredibly resilient. Why? Because to pull those molecules apart, you'd have to break all those tiny bonds at the exact same instant, which is just energetically almost impossible. The sum is so much greater than its parts. Okay, so we've got the strong, permanent bonds and the weak, dynamic interactions. Now for the really cool part. Let's see how these two systems work together in a couple of the most iconic molecules in all of biology. And we've got two perfect case studies to look at: the DNA double helix, which is life's information storage device, and the folded globular protein, which is life's all-purpose molecular machine. First up, DNA. Those long sugar phosphate backbones, that's the part that actually stores the genetic sequence, they're held together by those unbreakable covalent bonds. That's your permanent blueprint. But the two strands of the helix, they're zipped together by those weaker hydrogen bonds. And this design is just genius because it perfectly solves that paradox we talked about. It holds the code together securely for the long haul, but it also allows the strands to be easily unzipped for reading and for replication, all without ever damaging that permanent blueprint. Now, what about proteins? It's a similar story, but maybe even more dynamic. The main driving force that folds a protein up is that hydrophobic effect we mentioned. It buries all the oily, nonpolar parts deep inside, away from the surrounding water. And then this intricate network of hydrogen bonds, ionic bonds, and van der Waals interactions lock in and fine-tune that final, precise 3D shape that lets the protein actually do its job. If all this still feels a little bit complex, don't worry. Our source material offers one single, perfect analogy that just crystallizes the entire concept. It's this simple. Life is built with cement and Velcro. The covalent bonds are the cement, binding atoms together into these unbreakable molecular bricks for absolute permanence. And all those non-covalent interactions, they are like millions of tiny Velcro patches on the surface of those bricks. Individually, they're weak, but their collective power creates strong, specific, and, most importantly, reversible connections. This amazing strategy, combining a permanent framework with dynamic connections, it's not just a principle of biology, it's a fundamental principle of any sophisticated design. So as you go about your day, think about it. What else in our world is built this way? Where else can you spot the incredible power of cement and Velcro at work?